everyone. So I am going to start today's session with these things. First, I will repeat what we did in the last session, and then today's session. So first, we will do structure comparison. Means we will be downloading few structures, and we will be seeing how those two structures are similar or different. Just brief for today, and then we will be discussing a basic pathway so that we have understanding about how we can find some target on other pathways. I have kept it very simple so that everyone could understand this. Next, we will be visualizing how the selected target looks like with its ligand, means how a drug is interacting with the receptor. Then question answer session, and then assignment for you all. In the last session we had issue with downloading and installing Avogadro on many of our system. It is well known issue. I will try to find out the solution, and I will share with you, and before it is required I will share it with you. Next, I will repeat what we did in the last session. For that I will use Chimera X and Avogadro to show how these structures look like. In the last session we already downloaded a PDB file. Uh, Today I will download one more PDB file. I will go to RCSB again. I am going to basic site. Here I will search for some other molecule. uh, 53 isomer or something. Let us see what we get. I will download this simple structure. Here I am downloading PDB format. And it has been downloaded on my file system. In the last session, you all know that how you can open a file. In every software, you will find option of file menu. I guess everyone is able to see this file menu. This is menu bar. This is file menu, and by clicking this open option, you can select your file to open. I will use this option to open one file. I will select the file and click on open. Sometimes you don't need to navigate through other drives. You can directly copy paste your file path here in the file name dialog and directly open. So here is the structure, and you can also see the command. This is the command which it is using. So how you can use this command to open the file? Here you are seeing the command. You can copy it, and this is your command box. You can copy paste it here. So what I will do is I will first close the structure, close session. I will copy this path, copy selection, and then paste here. It is clear. I will paste here. I have pasted it and press enter. So like this I can open my file in this way. So this is the second method. Third one is to drag and drop. Fine. So this was your this kind of a simple assignment was given to you all last time. I hope it was clear to you all. I am audible to everyone, right?
Yes, sir, audible.
Yes, sir.
Okay, thank you. Next is drag and drop. So I will close this structure, select the file, go to my Chimera X and drop it here. Fine. So like that, there are multiple methods to open a file. These methods can be applied to almost all the software or packages. So at least you can use this approach to open file in any software, whether it is Maestro or any paid software or any other open source software. Similarly, I will demonstrate this with Avogadro as well. I will follow the same approach: file, open, or simply drag and drop. uh, File, discard. So like that you can visualize any molecule in any software. I will close it for now. So we will be going to next part. I will show you how we can visualize multiple molecules or PDF files. Like if you are trying to search for a single structure, there are multiple options available in PDB database. So here if we search again for P53 protein, there are multiple options like this one, this one. I will download these two files to open this file in multiple another tab so that I don't have to come back here. What I will do is I will press control button and then click here and then here. It will open in new tab. You will be able to see new tab here. I'm clicking it again. I will download these two structures. So this is my first structure. I will name it S1 or P53 S1. I will download a second structure as well. Now I have two structures. To open these structures, I have already told you how you can open single structure by dragging and dropping. Now to open these two structures, I will simply select these two structure here, and then drag it by keeping mouse clicked and drop it in Chimerax. So these two files will open. Now I will have a bit information about Chimerax interface. Here you can see this is models panel. Is it clearly visible? This is models panel. So you can see different structures are open. There is some ID thing. You will be using it later. This I is to toggle the visualization of the molecule. Means if I click on it, molecule will disappear if this is not checked. If it is checked, molecule is visual. I can click in this box as well. This will make my molecule selected, and to select multiple molecules I will have to use control button. Okay, so I will press save. So both of the molecules are hidden. I clicked here. So this is first structure S1 which I have downloaded, and this is second structure. Don't worry how your molecule is visualizing currently, just keep following me. And now you are not able to see whole the structure in this format. Here what I will do is I will use scroll button or to make it go back. Then I will click control sorry shift button, shift plus click to pan. Pan means Pan means I can move it anywhere, like this, left, right, top, bottom, or anywhere I can move this molecule by clicking on shift and click. I don't have to leave the click option because till we have mouse pressed, it will move with shift button. Okay, to rotate it, I will simply click, so I can rotate it on any side. Next is your how you are able to see this molecule. You need to use this option, molecular molecule display. Here you will get multiple options. Here is the complete toolbar. For now, you just need to use these four to five options given here. Using this option, I will also uncheck this selection for now, so that it applies on every molecule, so and every models here. We call protein structure a model in a software. So if I click on hide, it will hide everything. If I click on show, it will show up everything. So this is just it is to display the atoms, and in the protein structure there are multiple representation, and for nucleic acid there are different representation. In protein, ribbon structure is also present, so we can present protein by as a ribbon. So if I click on show, it will have it has got some changes, but it is currently not visible. What I will do is I will click hide here. I will hide these two options. This one is I have hidden, and this one, and selected this one. So this is my ribbon structure. As you can see, this is some helical structure. There is some seat which we represent by arrow, and this is using helix. So like this we can see two structures. Now what I will do is I will select one structure. You are able to see this green outline. So one structure is here and second structure is here. So these two structures are entirely different for now. But what we have done is we have selected structures are structures for P53. I will show how you can compare it in more sessions. So for now you can visualize that two protein structures you are seeing are looking bit different. I will close it here. Now I have one question. How do we have fever? Because it is something biological, like we have body temperature to fight for some defense against microbes or some infection. Can anyone tell me how this fever inflammation response works? Like what are the what is the process or something like that?
Sir, we can by increasing the temperature, the protein have the ability to denature itself in the high temperature. The the microbial proteins have the nature that the when the 38 degrees Celsius of the body temperature goes, they get denatured, and our body proteins resist to 40 degrees Celsius still.
So which protein denature at 37? Our own body's protein or
The viral protein is where the viral infection for the cold is occurring. The microbial proteins are specific.
okay, fine. So well, also how means how this process works. Like if I have some infection or viral infection, how this inflammation is triggered. Like there must be involvement of some molecules, proteins, or DNA, or some biological process. Like there is something happening in hypothalamus or brain which triggers further response, like it is generating some molecules which are generated, and later that raises the body temperature. So do you have any idea about that?
No, sir. I simply right now I learned that when the microbes enter to the body, the macrophages attach to them and engulf them, and after that the APC is formed for the immune response. After that, the body go to the inflammation pathway by the help of T helper cells. Uh,
Okay.
Other signaling pathway I don't know.
Okay, basically whatever immune response is there, biological process happen through biomolecules. Like if there is microbial infection, then there will be some response from brain which will be releasing some molecules, prostaglandins. Prostaglandin E, some E, and then in turn it so from hypothalamus this prostaglandin is released, and that elevates the body temperature by working with thermoregulation. Okay, so we have uh, 2 points, like there is hypothalamus, involvement of hypothalamus, and there is some thermoregulation kind of process. So we we take drugs to reduce this thermoregulation, means body to remove lower the body temperature. And how those drugs are called antipyretics. And how these antipyretics work, they control the enzymes or proteins which are released by hypothalamus, or or participate in this thermoregulation control. So to target those proteins, we need to know what are those proteins. Simply we can search on Google, like proteins involved in thermoregulation. There are research papers, and mostly we will be getting cyclooxygenase enzymes which are involved in thermoregulation process and targeted by antipyretics. So we can directly search for protein targeted by antipyretics to stop the fever. This
Excuse me, sir. Sir,
yes,
COX-1, COX-2, like are the basic prostaglandins which are responsible for thermoregulation in the fever condition.
Yeah. So we will be looking for COX-1 and COX-2. So we can get those structures because we know that COX-1 and COX-2 are targeted by antipyretics. Antipyretics drugs like uh,
Celecoxib.
celecoxib and many other drugs like paracetamol.
Yes, sir.
Okay.
Yes, sir.
So what we will be doing is we will be looking for the structure of cyclooxygenase here. Yeah, right. Cyclooxygenase.
Sir,
Crystal.
can you please repeat the
Hello. Is my internet breaking?
No, no,
No, sir.
it's
Actually,
okay, sir.
my internet is breaking. And the
Yeah.
answer you gave to the question, can you please repeat?
We had question. I was asking that how this and inflammatory response work, and then we use antipyretics to stop that inflammation in the body, and to lower the temperature we use antipyretics, and antipyretics target cyclooxygenase, which in turn target prostaglandins. Means not target but stop producing prostaglandins from the response of hypothalamus, and since these are targeted, these proteins cyclooxygenase is targeted by antipyretics, we can look for these structures. So by looking these structure we can also check for the drugs, how those drugs interact. Means if I have a structure of protein and some drug, then we can check how these drugs interact with each other. So first structure is here. Again I will download it like earlier. Do you have doubt? Do you still have the doubt? I don't know who was speaking.
No, sir. Clear.
Yeah, okay.
This time clear.
Actually, mostly I am focusing on this molecular structures, visualization in docking softwares. I'm not going in deeper with the processes and theory of or biology. I am just giving you hints so that you can go through Google or research papers to have more understanding about all those things. So basically, I had the problem that I don't know how antipyretics interact with inflammation pathway, and for that what I did, went to Google and I searched for uh, target of antipyretics. There I found that cyclooxygenase is targeted by antipyretic drug like fluorobuphene, yeah, ibuprofen, and other drugs. So I will get this structure, and here luckily I am getting this flurbiprofen. So I will download this structure. I will download it here and open it in Chimera X like earlier. Also, I can directly open the structure by just typing the ID. What I'm doing is I'm just trying it if I can open it, because there are multiple ways to open a single structure. I will just type the ID here instead of downloading. I'm clicking it, copying it, and pasting it here. Let us see what happens. I simply pasted the protein ID here. Is it visible to everyone?
Yes, sir.
Okay.
Visible.
So it is directly opened here, so I don't need to download it if it is present in protein data banks. Protein data bank. Most of the software which are able to visualize molecule, this should be able to directly open PDF file from online server of protein data bank. So this is our protein right here. I can again change its display type. I can hide it and show atoms. So it will appear bulky like this. I will hide everything again. I will show the this structure. So currently I am able to see complete protein here. There are other information as well. Now here I need to find our drug fluorovipan. So what I will do is there are other menus in menu bar. I will again go here and I will use these options, select or action, to select different structures out of this given structure. Also to zoom in or zoom out, you can just scroll. Fine. So I will click on select here and go to structure. Sorry, chains or residue. I will click on all non-standard. I will explain all those things gradually. Currently it is selected, but it is not displayed here because I have chosen proteins ribbon display, so that small molecules are not proteins, so they won't appear here. So I will click on so so all the other things which are not part of protein are now displayed. Using shift and drag to visualize it. Here should be multiple drug molecules. So just to select the drug molecules, again I will go to action, sorry, select residues, and here I am seeing FLP. So I am just selecting that thing. So there are, I guess, drugs are there, two drug molecules are there. I will change the structure using this option here, so that it has a bigger structure. I can visualize it clearly. So first structure is present here and second one is present here. So two drug molecules are present here. Okay, so now we can see that amino acids are like intermingled or acquiring some three-dimensional space here, and drug is trapped in between there. So this might be doing something which is not allowing this protein to work. Means this is not going to further convert prostaglandins or further produce prostaglandins, which after that, if prostaglandins are not uh, produced, then inflammation response is stopped. So the sitting of this drug molecule in this structure is causing some biological response in our favor. So I guess till now this is clear that what we have done, selected biological process.
Sir, how you selected the drugs?
I am telling you, I will repeat once again. First, see what I have done here. I have I have selected a biological process, then selected a target, and here we have selected protein. Okay, so and we know that how that protein is targeted. So here we selected antipyretic drug, searched in protein data bank for for that protein with a drug. There are other drugs which you will also find while scrolling down. So and then we have visualized it in Chimerax. Fine. So I will take one more example from PDB. I will go. I have searched here for cyclooxygenase, cyclooxygenase, and there we have got one structure, another structure, and then selectoxib and other molecules. And I will be taking one known example. Let me find one drug bank. Fine. Now I will search for antipyretic drugs. Pyretic drugs, and for antipyretic drugs there is for drugs there is a database, drug bank. I can go there. I have told about this database earlier, right? So here we can find lot of drugs. I'm just looking for a known example, paracetamol, or can anyone tell me more examples about antipyretic drugs?
Acetaminophenone be
Means
present.
in
Acetaminophenone.
general, means in daily day-to-day life, not this much complex. Simple, some simple example. Like this is ibuprofen, paracetamol should be there, right? Cyclooxygenase, paracetamol.
Sir, nimesulide. Nimesulide.
Nimesulide. Okay.
Yes, sir.
Okay. So I am taking this structure. What I will do is I will just copy it, and this is command line again. I am clicking on open and pasting that ID. I guess it is clear to everyone. I will close the old old structure by typing close and pressing enter. It is closed. I will I will type open and the ID of the protein and click enter. It should fetch the protein. This is the protein, and since there are different components in this protein, we can select different components using this select option. I will go to this select, then I will go to residues. There are non-standard residues and standard amino acids. Standard amino acids are part of the protein structure, and non-standard residues are not the part of protein structure, but they are like coupled with that. So I will select this nag or neem. I guess this should be there. I'm just guessing it right now. I will tell you how you can get all those things from the file. And also, if I see here residues, I will see this log information. This log, it is telling that this residue is for the drug. So I will tell
Excuse me, sir.
yes.
Sir, in the version of Chimera, like that, which is a older version, in that there is no like tab like log option. So like how will we
Yeah, you can
proceed?
see for log, like you can if you are not seeing any window, like if I am closing this model option or this log for now, in most of the versions you can find it under tool menu, or there I can see log is here, so I will display it. And I guess I have older version. No, I don't have older version for now. I will let you know how you can check for the log. Maybe by evening. I will try that. I will tell you that.
Okay, sir.
Okay. Otherwise, we have the traditional way that we can right click on the file, open it with Notepad. Simple. Why is Notepad open होने को time लगाता है? Okay, if I'm not able to open it in uh, Notepad, I will simply drag and drop it in. I will rename the file as .txt by appending .txt here. What I have done, what I have done here, apart from that PDB, I have used .txt. Like 3 and 8 is my file ID, file name, and .pdb is complete file name with the extension. I have appended .txt so that it is a text file now. And if I pose it as text file, I can drag it and open it in web browser. I will drop it here, and now it is open in web browser. Fine. So there is information about all the molecules here. So I will try to find NIM. What is NIM? Sorry, it doesn't have an NIM. I am opening the wrong file. Okay. First, I need to download that file for now. I will click on it, download it, and use .txt here to make it a text file. Download it and open it in browser. So this is an alternative if you don't have access to or like me, no, my notepad is hanging. So you can open it here like this and find NIM. So it should be here. So here we can see NIM is mentioned here. NIM name Nimesulide.
Nimesulide.
Actually, I am not directly giving you all the information. I am just I am doing all these things to let you struggle, and so that you try to find out maximum information out of that. I'm just telling you to open the file, and sometimes you don't find a way. You can do all these alternates, because information about biology and biomolecules is too much that you need to be capable of finding all those information yourself. So I'm just guessing it that nimesulide is here with NIM, and this same information will be shown in log file, or or in the X selection or some selection box. So if I go here, I will find out residues. There are BMA, BOG, and other things are there. I will try to find search for that as well. BOG. So it is some octyl beta glucose, some pyro, pyreno, pyrenosi- pyrenoside. So other molecules are also present, but for me, NIM is important for now. So to select that molecule, I will just click on select here, go to residue, go to name here, and click on it. It is now selected. You can see it by using this, seeing this gray line. You sorry, not gray. Yeah, this is green line you are seeing here. This green line. So I will change its visualization using this show option, so it is very bold and distinctly visible. I am able to see it right now. So I guess your question has been answered, that how I was able to select the drug molecule directly.
Yes, sir.
Okay, so I can see that these drug molecules are sitting within the protein structure, and and in the last option we also saw that uh, at some similar places other drugs were also interacting. So further we can calculate how these molecules are interacting, like hydrogen bond or salt bridges, or making some hydrophobic interactions. So all those things will be gradual, you will be knowing all those things gradually, but currently we can visualize that a protein has a ligand and it is visual to us.
So can you please tell me the command, how to write a command for like extracting particular protein from PDB?
Okay,
Can you
so
please tell me once again?
uh,
Only tell me the command.
yes, yes,
Just want to know.
so method 1 to open, method 2 to open files. First one is file menu. Click on open, select navigate, and select to the file. Second one is drag and drop. Third one is find the PDB ID, and in Chimera X command line type like this, open and PDB ID. Simple. So like this way it will open the file. Here is the command for you. You can opt for any option here, but I prefer this because I don't have to download the file. And one example for this is like open 3x18 something, means any random structure. I guess it is clear. I have answered your query, right?
Yeah, yes, sir.
Okay.
After writing open and then the PBD ID number and then enter.
Yes.
Okay, thank you, sir.
Well, so next we have already found that how molecules work. There are some natural molecules which we can find out, and for the same drug, like we have seen two drugs, and both of drugs were interacting near at the same place of the protein or cyclooxygenase. So for same purpose there are multiple drugs, so we call those drugs as derivative. There must be some natural compounds, like for antipyretics there may be some herb or some plant or some medicine, ayurvedic medicine, which might be working to prevent the fever or inflammation response. Next, to know more about uh, how these proteins are found, I am just naming you three databases. One is Uniprot. Here you will get all the information about protein structures, and this is pathway database KEGG, and how two proteins are interacting you can find in string database. I am just naming it. You don't need to worry about that. We will be including and explaining it in explaining it over that time. We have worked on it, and next one is we have already done this part, that we need to find a receptor. We have found cyclooxygenase here, and we we found receptor with its inhibitor. Inhibitor were two medicines which were inhibiting them and preventing the inflammation response. So we have visualized that. Just visualization is done, and we have learned one thing, that how we can zoom a structure or rotate a structure. If anyone has any problem, then they can ask me again. So this was for the day. If there is any question from anyone, please let me know. Or if you want me to repeat these things, let me know. Or if you want to do it right now and share the answer, share your response with me, let me know. I will wait for that.
Sir, I have one question.
Yeah.
Suppose if we want to study the particular protein is interacting with the receptor. Let's say this cyclooxygenase, for example, or I have a particular inhibitor for it. So, like the whatever the structures which are present in the PDB are with the some kind are already with the some kind of inhibitors. So,
Yeah.
what if I want naive cyclooxygenase structure? Then how will from where will I get it?
Means you want just cyclooxygenase structure
Yes,
without any ligand or without any interacting molecule, right?
yes, yes.
Okay. So the basic, the most simple thing is that you are seeing this is the text file, and you can
Yes.
edit it. Means you can delete lines or add lines in that notepad. So what you can do is you can try deleting some of the lines and see what is the difference in the structure in the visualization. Means delete this file, delete some of the lines, save it, and drag and drop in Chimera X. So you will understand that how this thing work. So there you will be understanding that first thing, that structure information is saved in the text file, uh, in PDB, we call it PDB format, and we can change that information by deleting or adding it. This is means basic thing. Later I will tell you how you can extract or add extract some of some part or molecule from the structure, or even add some parts in a structure, means complete molecule manipulation. So I will be explaining that. For now, for your exercise, you can delete some line for now from the file and visualize it. You will understand that how these things work. I will explain this pdb file structure and everything gradually. I am just keeping things very simple and easy for you, so that you learn, most of us learn it by very slowly, because computer part for biologist is like very hard to learn. Any other questions from anyone?
Sir,
Yes.
sir, I'm sharing my screen and I have an older version of Chimera.
Yeah, please share. I can see. I, yeah, please share.
Yes, sir, I'm sharing it. Now, sir, like tell me like how to select after selecting residue. Like you have a non-amino acid categorize, amino acid categorize, and non-amino acid categorize. In the non-amino acid categorize, you have a drug molecule
Yes.
and other whatever the bonds are there, and the amino acids are also there. So now, sir, how to select? See here, you have only the option for amino acid. See aliphatic, aromatic, charge, hydrophobic, negative, polar, positive, small, tiny. Sir, now how like differentiate? We can differentiate from amino acid and non-amino acid.
Okay. You are using residue option, right? Try
Yes, sir.
structure option.
Structure option.
What's in
Okay.
the structure option?
Backbone, ions,
Ligand. Click on ligand.
ligands.
You have not
Okay.
uploaded the structure.
Okay, sir. Okay, sir. Just I'm.
Fetch
Uh,
fetch by ID. Second option in file menu. Fetch by ID. Yeah. Type the PDB ID. Use 3 and X8. 3
three
N
x
N N like first
n
3 and 8 X. 3 and 8X.
three
3 and N
n
for no X. Sorry, 8X.
eight x.
X, yes.
Uh, After that,
Fetch.
fetching.
Yeah,
Fetch.
fetch.
I don't know what. I
What?
think something error. Chimera error.
What? Let me see the error. One.
Okay, sir. I'm just.
Yeah, open it. Yeah.
Oh no. Leave it. I'm searching. Where is it? Like I don't know. I have a open
Select any
and
protein structure. There must be some ligand or other structures.
open. Let's suppose this.
Yeah. So you should go to select.
Select residue or structure ligand.
Yep.
Sure, sir, can I click it on ligand?
Yeah. Click on ligand. So it should be selected. Is it showing any selection? Use scroll button. If you have
Scroll
ligand
button.
or not, then you can use what structure you are using. I just want to see it. See it. Means, can you tell me the structure name
Uh,
or
it
ID?
is yes, sir. I think I think I have to go again.
Go to tools. One thing. Go to tools.
Tools
Tools option. Can you see log or something?
log
Yes.
structure analysis sequence Let me select again, sir. Let me select again.
Yeah, select other protein because I guess this protein does
Sir,
not have
this,
ligand.
yeah, this one is 4D1, 4D1.
4D1
I think it's
L. Let us
L L
suppose that
dot PDB.
yeah, it is P53 from zebrafish is fine. So I guess
Yes,
it
sir.
doesn't have any ligand. It just protein structure is there. So I would suggest you to download some other structure which has some ligand so that you can visualize it.
Okay. So
You
should
can
I
try
do
it
now
once.
or sir? This one can shall I take this one IP5Z? I think in that you have a ligand.
What it has some ligand or not?
I think it doesn't have any ligand.
Okay, so
It is
uh,
complex with cytarabine. I
okay,
think with cytarabine.
okay, download it. It should work. What's that? Wait, send me a link first.
Sir, can we take this ID, just open and then type
Try
this
it.
ID number?
Yeah, try it once.
Okay. Now,
I would suggest you
installation.
always give a try, even if it doesn't work. At least you know what's the problem.
Yes, sir. Where should I type this
File.
command?
Um,
File,
sorry. It yeah. This has command option something.
close
Go to
session,
tools.
tools,
Yeah, I guess
tools.
command or something is there.
Here is journal command line. Yes,
Yeah,
command line.
click on command line. Can you double click on its maximize it so that you can see the command line? Yeah,
Yes, sir.
try
Yes,
this
sir.
option.
Open
S-
and then
Space.
paste it.
Are
Space
you?
is there.
Yeah. Space is there.
Okay, sir. Space. Then enter.
Yes. What
Error
it says?
during PBD
Error
fetch.
during PDB fetch. Error. What's version protocol? Error hello team. You should download it because there is some SSL kind of issue. Means it is a older version.
Yes, sir.
So
Yes, sir.
I don't know if it will work or not. It will not work because you need to give space between that part.
Okay, sir.
It's
Okay, sir.
problem due to software. Means things are upgrading our CSV, so is also upgraded, and it is might be using new technology. So older technology
Okay, sir.
has this issue.
Sir, can we do the drop and drag and
Yeah,
drop? I
drag and
think
drop. Yeah,
it is
try that.
possible.
Try that now. It
Okay,
should
sir.
work.
Mm, so from here to
You can direct.
drag, just command line now on the command line.
Not on the in the command line. You will get the path. Drop it in the black area.
Okay, okay.
It
I
is working.
think it's not working.
Not working.
No
Drag
issues.
it in
Just
command line once.
okay, okay. No.
Okay, you just open it using file option.
Okay, sir. Okay, sir. Mm,
File menu in the software.
copy. Sir, I'm copying on the desktop
I got you.
because it is easy for
Wait, wait,
like
wait,
extract.
wait. Open the Chimera X now. Open Chimera X.
Okay.
Sorry, Chimera.
Yes.
Go to File menu.
File menu.
Click on Open.
Open.
Paste. Delete this file name. Paste it once.
Okay.
Control V.
Let me
No, no. Just click. Press Control V there.
control V.
Yeah.
No,
It's not
not.
working.
No, no, it's not working. Not working.
Okay, okay.
I have to paste on the desktop only. I
Yeah,
think
yeah.
so.
Okay, you can navigate it on that because
Paste it. No. Yeah. Yes, sir. It's pasted here. IP5Z.
your system is in a
IP.
32-bit something, right?
Yes, sir. Yes, sir. 32 bits.
Okay.
This is 1P5Z open. Yeah. Now it
Yeah,
has some
it is
ligand.
open.
Okay. Now we have to go to select residue or structure, the ligand. Yes, sir.
Yes. Try. Don't worry if
Yeah.
it is wrong or right.
Okay. So
Whatever
it's
you
like
see.
something highlighting. There is
Yes.
something
Now
highlighting.
go to action or preset something. There might be you will see that option as well. Ribbon, not ribbon.
Yeah,
Surface.
ribbon.
Go to surface.
Surface. Okay,
Yeah,
surface.
like click on show,
Click on
show,
show. Okay,
show
show.
first option.
Yeah,
So
show.
I those options were available in the ribbon in Chimera X, and we need to select it from a drop down menu or menu bar.
I think action, na sir. From action, you have to surface and
Yes,
then
surface.
show.
Yep. If you see Chimera X here in the Chimera X, there you are seeing atoms, cartoons, and surfaces.
Yes, sir.
These names should be present in your drop down or older versions. So
Yes, sir.
like that you can use find something similar in your package of software. Means any other software, those will be having similar options.
Okay, sir. Okay, basically, like I have to, like I have a challenge to find out the same thing in Chimera also.
Yeah, if you don't find that, just go to Google, type Chimera, show surface. So like that, there will be tutorial or some official documentation for you. I will also make you walk through official documentation of any software so that you don't have to struggle and you will be finding solutions yourself.
Okay, sir. One thing more, sir. Like, can we replace this ligand with other drug molecule also?
Yes, we
Okay,
will
so that
learn
is
it
like
later. We
okay.
will learn it because that's the part of docking that we have one protein and how other drug is going to interact. That is
Okay,
ultimately
sir.
going to replace older molecule and will be replacing older molecule and will be interacting with that protein.
New molecule, and
Yes.
after that we will find the docking score.
Yeah, that's part of docking.
Okay. Okay. Thank you, sir.
Okay.
Okay, got it,
Is
sir.
there any other question from anyone else so that I can just move to assignment part?
No question, sir.
Okay, if anyone has any question, just drop me in Telegram group so we can have random schedule as well and discuss those problems. So for your assignment, you need to do yeah, you people have selected one research article for related to docking and dynamics. You find what protein or structure they have used. They might be using providing some PDB ID which they have used for their purpose. Find that ID or find that name and search in PDB file, PDB database, and open and visualize it. And if that molecule was already having some ligand or ligand, so you need to highlight that like we have done in today's session. Next, Select a random biological pathway or any pathway of your interest. Then find some protein and try to find a protein which has some ligand and highlight that. Both of the activities are similar. You can combine them. It's your choice. So it should take maximum 10 minutes for you to do these two parts, and try to send me it in PDF format. This is also a requirement for today, because I will be also helping you how you can make present things properly and easily. One part was missing. Okay, there is nothing. Tomorrow I will be discussing about receptor and ligand interaction, means how the one receptor is interacting with a ligand. So mostly that will be kind of how hydrogen bonds are forming between two residues or two molecules, something like that. So, so I am ending this session now. If anyone has any question, so drop it in on Telegram. I would suggest you to complete today's assignment, means by giving 10 to 20 minutes till tomorrow, so that you have a better understanding what we are going to discuss tomorrow. Uh, Otherwise, if you have any problem, let me know. I may not be solving immediately, but I will try to find solution quick.